Thank you very much. And I guess if Jerry was the token humanized mouse presentation, mine is the token natural host presentation. As Jeff said, in Africa, there are about 50 species of non-human primates that are naturally infected with their own species-specific version of SIV. It is clear that these animals were the sources of the viruses that caused HIV infection in humans. SIV-C. Am I okay? Oh. All right. So, should I carry on? Acapella? Yeah. Yeah, this is going to go downhill real fast. HIV-1 coming from cross-species transmission of SIV-CPZ, which was a virus that naturally infected chimpanzees. HIV-2 coming from cross-species transmission of SIV-SMM, which is a virus that naturally infects Sudi mangabees in Western Africa. But luckily for us, now we don't have the slide. Luckily for us, the viruses that the SIV-SMM strain causes progressive infection in Asian non-human primates. And so one of the questions that these animals in Africa have been co-evolving with their own species-specific version of SIV for quite a long period of time, but importantly, they've been co-evolving to the point where they avoid disease progression. So natural SIV infection of non-human primates in Africa does not result in progression to simian AIDS. The timeline that this coevolution has happened has been a topic of much discussion about the advancing isn't working either. Okay. So the timeline has been of a considerable amount of discussion. I think some of the strongest data that suggest the age of SIV come from work from Preston Marks' group, where they were able to find that there was species of SIV on this island called Bioko off of the coast of the western coast of Africa, and they could find several different strains of SIV. There are drill monkeys on Bioko that have SIV, and so they were trying to argue that the geologic age of the Bioko island kind of gives an indication as the minimum age of SIV, and they kind of argue that SIV is about 32,000 years old. And so we are interested in studying the natural host of SIV because we can do comparative studies and try and understand the aspects of the natural host virus infection compared to the non-natural host of pathogenic infections to try and get an understanding of what aspects of the infection are important for disease progression. And so one of the first things that we know is that the natural hosts have not co-evolved to protect against virologic or to control plasma viral load. An SIV-infected natural host will have a viral load that is comparable to an HIV-infected human that is ARV naive or an SIV-infected rhesus macaque. And so this is just kind of a list of some of the key aspects of progressive infection that we think of as being important for progressive infection. Depletion of mucosal CD4 positive T cells routinely happens in both HIV-infected humans and SIV-infected Asian macaques. You get some degree of CD4 T cell loss in mucosal sites of the natural host, but loss of CD4 T cells from peripheral blood is very rare. Other pathologies that happen within the gastrointestinal tract seem to be absent in the natural host of SIV. You do not get preferential loss of IL-17 producing CD4 T cells or CD8 T cells or the type 3 ILCs. You don't get systemic inflammation in non-progressive hosts. They don't get microbial translocation, and of course, they don't succumb to fulminant AIDS. And I've got a different screen here, and I'm not sure how I'm going to advance the slide. Carefully. Oh, yeah, I did the bad thing. I can see it here, and I can advance it. So, okay, now this is working. This is intermittent. And so about 10 years ago, we started trying to understand immunological abnormalities that might be present in natural hosts for SIV compared to non-natural hosts. And the animals that we studied were African green monkeys. As Jeff said, this is the most commonly used model for non-progressive infection. And there are about 6 species of African green monkeys. The ones that we are studying were imported from Tanzania about 15 to 20 years ago. They're called Chlorocebus pygaraphus. Most people in the field are studying animals that are called sebaceous animals that were transplanted from slave ships probably onto St. Kitts Island in the Caribbean. And similar to what was discussed with the Mauritian cynomolgus macaques, the Chlorocebus sebaceous animals have a very small founder population, so they're pretty genetically similar. But one of the things that we were kind of surprised by, and this was true whether we were looking at any of the species of African green monkeys that we were able to get our hands in, and you could also see it in Erythrocebus patus monkeys, was that if you look in peripheral blood at T cells, and this is CD4 by CD8, we were really surprised to see a very low frequency of T cells that express CD4. In this adult African green monkey, only 5% of the T cells were CD4 positive. They had a huge population of T cells that expressed the alpha chain. This is auto-advancing now. The alpha chain of CD8 without the beta chain. And what we were able to show is that many of these CD8 alpha T cells were originally CD4 positive T cells. So the reason that there were very few CD4 positive T cells in the peripheral blood of this adult animal was because they had actually downregulated CD4, upregulated the alpha chain of CD8. And these cells could do every effector function of a CD4 positive T cell, including maintaining restriction by MHC class II. But because they don't express CD4, they were resistant to SIV infection in vivo. And so to assess this, we flow cytometrically sorted naive CD4 T cells, memory CD4 positive T cells from adult African green monkeys, these memory CD8 alpha T cells that were initially CD4 positive, the canonical CD8 T cells, monocytes, and double negative T cells. And so the open circles indicate times when we could amplify SIV AGM DNA from these different populations of cells. And you can see that we routinely were able to identify SIV AGM within the memory CD4 positive T cells. The closed circles are where we had no detectable viral load, and we're basing it on the lower limit of detection. So when we looked at CD8 alpha T cells, only in a few animals did we find any SIV DNA, and you can see that they were at very, very low levels. So it seemed like downregulation of CD4 with immunological preservation of function might be a mechanism by which the animals were avoiding disease progression. So to follow up on these studies, what we were interested in trying to understand is the mechanisms underlying CD4 downregulation. And by looking at different cross-sectional populations of African green monkeys, we were able to kind of conclude that immunological experience was important for CD4 downregulation. So Vanessa Hirsch, our collaborator on this project, had been breeding African green monkeys. These are, again, from Tanzania, so it's easier to breed them than to keep importing them. And what we could see is in that infant African green monkeys, they had CD4 frequencies that were comparable to HIV uninfected humans or SIV uninfected Asian macaques. So this would suggest that the mechanism underlying CD4 downregulation wasn't thymically, but it was immunological experience systemically. Consistent with this, we had one animal that was quite geriatric. When it was imported from Tanzania about 20 years ago, was SIV positive, had a viral load in the 100,000 range, and over the course of the past 20 years had accumulated more and more cells that had downregulated CD4 to the point where its CD4 count is 0. It no longer has detectable SIV viremia. So it looks like, at least functionally, this animal has cured itself by downregulating CD4 from all of the CD4 positive T cells. So even though this animal didn't have detectable CD4 positive T cells, it could still respond to neoantigenic stimulation. So this is an MHC class II restricted response to a vaccine that we gave the animal. The animal had never seen that antigen before, and it could still make a response out of this population of cells that had downregulated CD4. So that suggested to us that the immunological experience didn't have to be through the T cell receptor, and that any type of maybe proliferation systemically could result in CD4 downregulation. And in fact, if we flow cytometrically sort CD4 positive T cells or CD4 negative T cells from African green monkeys and then feed them homeostatic cytokines that are important for T cell proliferation, and this includes IL-7, IL-2, and IL-15, you can see that homeostatic proliferation by itself is sufficient to cause downregulation of CD4. And so next we were curious about the molecular mechanisms responsible for CD4 downregulation. And so to address this, we flow cytometrically sorted cells that were in the process of CD4 downregulation And we did these experiments in 2 species of animals that have CD4 downregulation, the African green monkeys and the potus. And for comparison, we sorted cells that had been given the same stimulant but not downregulating CD4, and that was from the rhesus macaque. We then extracted RNAs and looked at the differentially expressed genes. So we particularly targeted genes that were expressed similarly between the African greens and the potus, both animals who have downregulation of CD4, but different than the rhesus macaques that proliferate without downregulating CD4. And there were about 2,000 differentially expressed genes that fit that category. And I just want to point out that CD4, I think, was the 10th differentially expressed gene being downregulated in the African greens and potus. CD8 alpha was also in that list, being upregulated in the AGM and potus. And then we functionally annotated those differentially expressed genes. And one of the pathways that lit up was epigenetic modulation, and in particular, DNA methylation machinery. And there was one enzyme in particularly that stood out. It's called TET3, which stands for 1011 translocation enzyme, which is important for removing methylation signals on cytosines. So it makes a gene more expressed if the methyl group is taken off. And it turns out that there's a chemotherapeutic agent called 5-AZA2. It also goes by decitabine, which inhibits addition of methyl groups to cytosines. And so here we're looking at African green monkey CD4 T cells stimulated through the T cell receptor with anti-CD3 and CD28, and you can see significant amounts of downregulation of CD4 as the cells divide. But when we inhibit addition of methyl groups to cytosines, you can see we can still get copious amounts of proliferation, but less downregulation of CD4, and that was statistically significant across each of the different generations of division, from cells that don't divide to cells that divide 6 times. So if the prediction is that inhibition of removing methylation groups is important for the downregulation of CD4, the prediction would be that there are accumulations of methylation in the DNA of the cells that downregulate CD4. So to start out with these experiments, we designed primer sets that kind of spanned different segments of the CD4 gene that are thought to be important for expression of CD4. So this is the transcription start site of CD4. Upstream, there is an enhancer of the CD4 gene, which is thought to be important for CD4 expression in the thymus. Downstream of the transcription start site, there's something called the CD4 silencer. And then more recently, there is a maturity enhancer within the CD4 gene that is thought to be important for maintenance of CD4 expression once the cell leaves the thymus. And so we developed primers against different regulatory regions of the CD4 gene, and then we do a technique called bisulfite sequencing, which tells us whether or not cytosines have methyl groups or not. Each open circle is a clone that had no methylation, and the closed circles are clones that did have methylation in particular segments. And so we're just looking here at this maturation enhancer within the CD4 gene, and you can see that within CD4 T cells, remember there are some cells that have not yet downregulated CD4, we only found 2 clones that were methylated. In the CD8 alpha T cells, these are cells that used to be CD4 positive but have subsequently downregulated CD4, you can see significantly more clones that had methylation signatures. And here for comparison are the canonical CD8 positive T cells. And so we did this in targeted regions. You could see that the place where we saw differential methylation was really in genic regions within the CD4 gene after the transcription start site. So to follow up on this and try to take a more global view of the CD4 gene methylation status, in collaboration with Danny Duick, we did next generation sequencing of the entire CD4 gene, all of the exons, all of the introns, all of the regulatory elements, and we even went upstream and downstream of the CD4 gene. And so the only difference here with the bisulfite sequencing is whether or not particular cytosines are methylated. So in red are the CD4 positive T cells, which are clustering away from the CD8 alpha T cells, which were originally CD4 positive, who are clustering more with the CD8 alpha beta T cells. And when we look across the gene and ask how is that methylation differentially occurring, you can see that in the green are the CD4 negative CD8 alpha T cells, the blue are the canonical CD8 positive T cells, the alpha beta T cells, and in red are the CD4 positive T cells. The shadowed areas are where differential methylation occurs, And you can see it's always the case that the green, which are the old CD4 positive T cells that have downregulated CD4, are having increased methylation similar to the CD8 alpha beta T cells. And here is that maturation enhancer like I talked about earlier. And again, using the next generation sequencing approach, we see the same thing that we saw by the targeted sequencing using clones. So to wrap this up, African green monkeys live healthy lifespan irrespective of their SIV AGM infection status. African green CD4 positive T cells become CD4 negative by downregulation of mRNA for CD4. The resulting cells are resistant to SIV infection in vivo, but can still do everything a CD4 positive T cell can do. The downregulation of CD4 occurs concomitantly with downregulation of this demethylation enzyme called TET3, and regulatory regions as a result of that of the CD4 gene are differentially methylated in the CD4 negative CD8 alpha alpha T cells, and we can therapeutically block this, at least in vitro, with decitabine, and that results in maintenance of CD4 expression. So overall, the conclusion might be that coevolution of African green monkeys with SIV AGMs seems to involve downregulation of CD4 by epigenetic modulation of the CD4 locus. And we think that this might lead to novel therapeutic interventions to either downregulate TET3 in humans, but of course first we would look in non-human primates, the rhesus macaques, or in inhibiting TET3. And so I'll just close there and thank the individuals who were important for this work. These studies were done by a very talented postdoc, J.C. Mudd, who's just recently had a perfect score on his K grant, so he's on the market looking for a tenure track investigator position. And all of these studies have been done in collaboration with Vanessa Hirsch, who imported these animals from Tanzania a long time ago. And the RNA-seq was done in collaboration with Mark Cameron, and the bisulfite sequencing using the next generation approach was done in collaboration with Danny. And I'll thank you for your time and attention and take any questions.
Don't forget to announce yourself.
That's right. I'm Mario. It's great data, Jason. I'm just curious how you explain the lack of difference in viral load in the animals with so few CD4 cells.
Yeah. So Guido had a study 7 or 8 years ago where they had done CD4 depletion in sooty mangabees, and I think the conclusion was that it didn't take very many CD4 positive T cells to maintain a very high viral load. And we do see that when animals become extremely, well, they've converted most of their CD4 cells, as is the case with the animal who cured itself, that their viral loads do get pretty low. But the gist is that it doesn't take very many CD4 T cells to have a fairly high viral load. I mean, and high is maybe 10,000. We're not talking millions like you see with a rhesus macaque.
Are there any other questions for Jason? I guess controversially, in following up on Mario's question, any role for infection of myeloid cells in that setting?
Not that we have been able to see.
Thank you.
And the fact that that animal has plenty of macrophages, and CD4 does not seem to be lost by myeloid cells in African greens like the CD4 positive T cells, but that animal had no viral load. So if there were plenty of macrophages that could have supported viral replication, we just didn't see it. Maybe SIV AGM doesn't like to infect macrophages either. That's another possibility.